Okay, so I'm going to change paths a little bit. I'm going to talking about a collaboration that I have with the Department of Engineering to design a tissue organoid chip to model RPE chord interactions. So the talk is structured in 4 different parts. I'm going to first describe the model itself, and then I'll show you some experiments we did with primary RPE cells, and then move on to some RPE cells derived from stem cells from normal donors, and then I'll show you some interesting results that we got from RPE cells derived from donors of stem cells from donors with AMD. So why even study or sort of why even make a model for macular degeneration? Well, it turns out the mouse hasn't been very kind in terms of giving us great insight into human AMD. Also, this article that came out last year in Nature, a suggestion by the FDA, that we should begin to explore other models for human disease. And one of their suggestions was to begin studying stem cells, which hopefully could be engineered into tissue chips or organoid chips to eventually study new drugs and other interventions for human disorders. So it's my opinion that with the use of single-cell genomics and using a model such as this, we should be able to address which cell types are selectively vulnerable or resistant in macular degeneration, answer whether AMD progression can be defined as an abnormal aging trajectory, and identify the molecular pathways perturbed in each cell type, as well as identify new cellular and molecular targets for intervention. So the model that we're using has 3 components. It has a top layer, which is made of a polydimethyl siloxane material, which is then overlays a porous membrane, which then has a bottom layer, which has, pointer is not working. Oh boy. All right. Let me see this. It has some channels in it, as you can see here, and some wells in the bottom layer. This is the model, the apparatus with red dye in it, and I think you can see the wells filled with dye, as well as the channels which contain the dye, showing you how they're all interconnected. This is a cross-section through a particular well in the model, and you can see that there's a top layer, there's a porous membrane, and there are channels below it. This is the bottom chamber, and it has steps between it. So the way we inject the material is that we have a hydrogel material in which we inject primary endothelial cells and fibroblasts into the middle chamber. We allow this to grow for 7 days, and you can begin to see the vasculature forming in this bottom layer. We then inject endothelial cells into the 2 outer chambers and overlay a layer of RPE cells, primary RPE cells for this particular experiment. We wait 10 days, and the RPE layer is confluent, and we also have a very mature vasculature underneath. This is a confocal image showing that, in fact, the overlying RPE cell is expressing RPE65, which is a specific marker for RPE, and also the underlying vasculature is expressing CD31, which is a specific endothelial cell marker. What we found is that it takes time for the vasculature to mature. The density is present. There is some vessels at day 3, but we don't consider them to be very mature. And it takes a period of at least 21 days to get a very mature vasculature in that bottom chamber. And while you may have vasculature, really the purpose of this experiment was to determine if these vessels could be perfused. So what we did was, to show that initially, we injected dye, 70 kilodalton dye in one of the channels, and with a little positive pressure, after 5 seconds, you can see the dye filling some of the vessels, and after 30 seconds, it's a lot more filling the vessels. And a cross-section of that particular chamber, you can see that, in fact, the red dye is filling these vessels very nicely, and you have staining of the endothelial cells with have DAPI staining, and you have CD31 staining of the endothelial cells. So, as we all know, the choroidal vasculature does not have tight junctions like the retina, so you would expect to hopefully see some kind of diffusion. There wasn't very much for the 70 kilodalton molecule, so we then injected 10 kilodalton molecule, and you can see that after 10 minutes there's some diffusion of this smaller molecule from the microengineered vessels. After 60 minutes there was a lot more. So there is definitely diffusion of smaller molecules, and it's very steep for the first 20 minutes and sort of plateaus out at 60 minutes. So here is a video, it's working, good, of the injection of the dye. Should be, yeah, here we go. It's the injecting of the dye on one of the channels with a little positive pressure. You can see it perfusing through the vasculature. So this was very encouraging to us because now we can sort of study perfusion. And this is the same principle with microbeads, fluorescent microbeads. You can see it very nicely flowing through the vessel. So this is really exciting to us. Okay, so the next question is, what in this model that we have, is there an influence of the overlying RPE to the vasculature? And also, does the vasculature influence the RPE in any way? So we did find that, in fact, if we grew the vasculature in what we call a co-culture situation, which means vessels plus overlying RPE, versus monoculture, which is just the vascular alone, that in fact the co-culture situation caused the vasculature to be much more dense. And you can see the density increased here at day 7, and day 14 it's even more significant, and at day 21 also. So this would probably be expected because RPE secretes VEGF, so you would assume that this would be the case, but it shows that there's crosstalk between the overlying RPE and the underlying vasculature. But what about the RPE? Does this underlying vasculature influence the RPE? Well, it does. Again, these are primary RPE cells. The blue staining is DAPI for nuclei, and you can see that when we stained for melanin, melanosomes, that there was an increase in immunofluorescence for the melanin content in the co-culture situation versus the monoculture. It was very significant. Laminin secretion was increased under the co-culture environment versus monoculture. Laminin is a protein secreted by the RPE and deposited into the basal lamina. And the staining, the tight junction staining was different between co-culture versus monoculture. The staining for the Z01 protein was very distinct and regular, and it was not very distinct for the monoculture situation. So that would suggest that there might be a barrier disruption if you grew the RPE cells by themselves. And what about drusen? Well, it turns out in the co-culture situation, there was number of drusen was much more. It was very significant compared to the monoculture. And phagocytosis. So what we do is we feed the apical layer of the RPE with beads, which are have bacterial beads, and when they get ingested inside the cell, they fluoresce because of the difference in pH. And you can see that under the co-culture situation, there was much more phagocytosis as opposed to the monoculture. It's very significant. Okay, so the next step is, so this is great. This works with primary RPE cells. We can probably get a lot of information, but RPE cells have limited longevity. So we then got RPE cells, which were differentiated from stem cells from a normal donor. And this is sort of a quick image of that. You can see that these iPSCs have very regular borders, and they're actually ingesting these bacterial labeled beads. When we then said, okay, let's ask the question, does the RPE derived from iPSCs any different from the primary RPE? And you can see with respect to melanin content, there's no significant difference. There was no significant difference with respect to drusen, number of drusen. And there was no difference with respect to vascular density. So then we got a few AMD donors, took their stem cells and differentiated them to RPEs. And just looking at the surface layer with a microscope, bright field, you can see that there's a difference in pigmentation between an iPSC from a normal donor versus an AMD donor. And dramatically, the melanin content was significantly different from the RPE derived from the AM donor versus the normal donor. It was really decreased. And so was laminin content. Laminin from the AMD donor was much less than the normal. And when we looked at the number of drusen, the AMD donor, as you might expect, had much more drusen frequency than the normal, and the diameter was also much larger. Then we said, well, let's try to see what perfusion does. So we decreased the flow rate that we normally use in our model from 100 microliters to 60 microliters per hour, and we found that, in fact, the number of drusen increased, but the diameter of the drusen did not. So that was interesting. We then compared the Z01 staining, the tight junction staining, and we saw that, in fact, the AMD RPE cells had very irregular staining for the tight junctions compared to the normal RPE. And we then added fluorescein to the apical portion of the RPE, and we measured diffusion of the fluorescein across the RPE layer into the underlying layer. And you can see that in the AMD RPE, the diffusion of fluorescein is twice as great as the normal donor. So there's a tight barrier. The barrier is actually disrupted in the AMD RPE cells. And what about phagocytic activity? Well, this was really dramatic. The RPE cells from the AMD donors had very little phagocytosis compared to normal. We measured this at day 7. You can see it's almost barely above baseline from the AMD donor, and 14 is a little bit more. And at day 21, the phagocytosis was significantly decreased compared to the normal donor. Okay, so finally, we all know that cigarette smoking is the biggest risk factor for age-related macular degeneration. And Stuart's saying yes, so I know it's right. So we made smoke extract, and we injected the smoke into the side chambers of our model. And what happens here is that the smoke extract gets perfused through the vessels. And the question is, what does this smoke extract that's perfusing through our microengineered vessels have on the overlying RPE? We did not put smoke extract on the RPE at all. And we were interested at the moment in complement, so we measured some different complement components. One of them is C3, which is called complement 3. And we saw that with 0% smoke extract versus 1% versus 5%, there really was no difference in the amount of complement 3 based on immunofluorescence. In fact, it was comparable to the AMD donor, which was not even exposed to cigarette smoke extract. We then looked at the complement factor H protein. And you can see that as the smoke extract increased from 1 to 5%, even from 0 to 1%, there's a dramatic decrease in the amount of complement factor H made by the RPE cell. And we then looked at the MAC complex, which is the terminal component of the complement pathway. And we didn't see much until we got to 5% smoke extract in terms of the immunofluorescence for this protein complex. And it was quite similar to the AMD donor, which had not been exposed to smoke extract. Okay, I'm going to rush through this. We had further experiments on AMD RPE cells and normal RPE cells derived from a normal donor. And the number of drusen's significantly increased as we increased our smoke extract. So smoking does increase the number of drusen, at least in our model. And finally, what did it do to phagocytosis? This was very interesting, that as we exposed the RPE cells to more and more smoke extract, look what's happening to our phagocytosis. It's decreasing. It really, and the AMD RPE, which had no exposure to smoke extract, was very low in terms of its ability to phagocytose. So let me end up thanking the people in my lab. Special thanks go to this wonderful, wonderful graduate student who I had the honor of mentoring. This was her PhD, plus she also made a model for the intestine, an organoid model. Thank you, and I'll take any questions.
Well, thank you, Dwight, for presenting this exciting new model. Questions, please.
Yes.
Catherine.
Really beautiful model.
I'm gonna get up.
Can you put in things like glucose into the media and see if you can change the artificial vessel formation as a model, for example, of diabetic retinopathy?
Very good. Future experiments. We actually applied for a grant for this, so we're waiting to find out what happens. It's very expensive to do this kind of work.
Claire.
Again, fabulous model. What happens as it ages? How long can you grow this combination of cells for? Which cells drop out first, and how do you keep them going for longer?
Stay tuned. Very good question. We didn't go much out beyond 21 days, so I understand what you're asking, and I think that's another question. I think the idea with this model is to really, because I think you'll agree that, in fact, the choroid is now believed to be where the AMD actually starts. So I was really interested in this choriocapillaris dropout in early AMD. So the question is, if you decrease blood flow in this model, what do we expect the RPE to do? And also we can add cells to this model. We can add mast cells, lymphocytes, macrophages, which exist in the choroid, and see how each cell is acting in response to an insult. And we can actually tease apart what each cell is doing.
Ken.
Dwight, again, great model. Can you comment on the fluid dynamics? How fast are things perfusing through your vessels?
Yeah, so I had that up there. So normally, okay, so up there it's 100 microliters per hour, and then we decreased the flow rate to 60, and then we saw that increase in drusen.
So Dwight, there's an age-old debate about whether drusen come from the choriocapillaris, proteins in the plasma, versus from the RPE. Is there, so you're finding ApoE in your drusen, is there ApoE in the material that you're circulating into the choriocapillaris? Could it be produced there, or is it being produced by the RPE?
No, it's just tissue fluid. Yeah, there's no.
So you think it's is the RPE making the ApoE that you're detecting there?
Yes. In fact, I think I showed you a slide where the drusen is just underneath the RPE.
Have you cultured the RPE without the choroid and seen the ApoE?
Yes. We did that. That was one of my experiments.
Fantastic.
So without it, you don't get as many drusen. Yes.
Okay, thank you. Another hand for Dwight, please.